Hello everyone. In this video I'll show you how you can upload your single-cell RNA-seq data such that we can run our tools to reconstruct a bonsai tree representation of the data that you can then explore in the Bonsai-scout app. First of all, you go to this website where you can already see that there's some example results available of datasets in which we already reconstructed a bonsai tree, and you can explore this tree and see if you like that. Most importantly, it has a data upload section, and I will just quickly show you how to upload your data. Now you can choose between uploading a TSV file or an mtx file, which is a sparse format. Both are not incredibly complicated, especially because we have information for what you have to upload in different tabs. I have a TSV file, so I'll just quickly show you that. I already filled in my email address and project name. Now these are very handy to fill in, but not actually necessary, but I do recommend to do it. So for the TSV file that you have to upload, it should look like this. So we have columns separated by tabs. First column just containing the gene IDs, and the second column, or the first row, contains the cell IDs. And then in the different entries of the matrix, you will just see the number of mRNAs for that gene in that cell. So I already prepared a matrix for just 64 cells to have a very quick and small example. And then the last thing that you can upload is annotation. This annotation will be used to show you this annotation on the tree when you explore the data. It can be very useful. I already prepared something on that. So the annotation file should look like this. It can be in TSV format or CSV format. And it has different columns again. In the first column we have the cell IDs, and in the next columns we first have, well, the type of the annotation that you upload, and then for every cell what its value for this annotation is. You can do categorical variables and numerical variables, and the Bonsai-scout will automatically recognize that it puts numerical values on a sequential color map. If you have several annotation files, you can also just copy them in a folder and zip the folder and upload that. Then we'll just go over all the folders and add all the annotation that is in there together. Now, last thing that is possible is to upload feature values, so additional things that you measured on these cells but are not RNA-seq data. For example, maybe you want to also show the UMI counts, the raw counts on the tree for different genes, or you've measured other things on the cells. Then you can upload these as matrix files, but for that please look at the README in the Bonsai GitHub. It will explain exactly what you have to do. So I already uploaded my annotation, and then I just click submit, and this is all you need to start the Bonsai pipeline. Now you can choose either to store this page address, and then you can always go back and see if your results are already done, but it's most convenient to give your email address before, and then you'll just get an email when it's done with the links to the results. I already did this, so I will go to the email that I got. So you get two links. One leads you to the Bonsai-scout, which is the app to explore the data, and the other one goes to a download page where you can download the results of all the tools that we ran for you. The Bonsai-scout looks like this. You see that my small data set resulted in a beautiful tree that we can now look at in different ways. But for how this Bonsai-scout app works, we have made two other videos that you can look at. I'll not focus on that. The other link that I have is going to the download page, and there you see that we get sanity output. These are the normalized expression values. Cell state output, which gives clustering of cells into statistically indistinguishable groups, and the Bonsai output, which gives you the trees, but also the posterior on the gene expression states at every node of your tree. So I hope that kind of makes it clear how to upload data to our server. If you have questions, don't hesitate to contact us. We'll be happy to help.